Hello, my name is Sanoja, and today I'll be talking through the structure of the phospholipid bilayer. So the phospholipid bilayer is what forms cell membranes, and they control what goes into the cell and what can leave the cell. So membranes in the cytoplasm of eukaryotic cells actually separate them into certain compartments, and the entire structure itself is only around 7 to 10 nanometers in width. So they're very small, and you can only see them through a transition electron microscope, or a TEM, with a magnification of around 100,000. And with this microscope, you'll see around two lines separated by a small gap, and this is the phospholipid structure. So if we take a closer look at the structure itself, we can see that it is made up of a hydrophilic head, which is polar. So hydro meaning water and philic meaning loving. So it is a water-loving head. So it faces either the cell exterior or the cytoplasm. And on the other hand, we have a hydrophobic tail, which is non-polar. And in between the tails are weak hydrophobic interactions. And so together they form the phospholipid bilayer. So they are what separate the cytoplasm and the cell exterior. And this entire structure itself is actually a partially permeable membrane. And what we mean by that is that it is permeable to non-polar molecules such as oxygen and fatty acids. And it is also permeable to small polar molecules such as water, ethanol, and carbon dioxide. And on the other hand, we have the structure that it is impermeable to. So it is impermeable to ions and large polar molecules such as sugars and amino acids. So how do you think these large molecules that cannot just freely pass through the membrane get inside the membrane? So the answer to that is they actually use carrier proteins or channel proteins. So carrier or channel proteins, and these are actually a transmembrane structure. So what we mean by transmembrane is that it spans the entire membrane, so from the exit to the entry of the cell. And so the large structures, if they have the correct receptor, they can bind to these carrier or channel proteins with the corresponding receptor, and once bound, they can easily go inside the cell or leave the cell using this structure and not having to pass through on its own because it will be impermeable anyway. So on the other side, we have something called cholesterol. So cholesterol is found in between the hydrophobic tails of the phospholipid. And what cholesterol does, it basically acts as a stabilizer, and when temperatures are too low, it prevents the solidification of the phospholipid. So it prevents that from solidifying, because if it's solid or very rigid, it will be hard for substances to go in and out, and it will be hard for the entire structure to be fluid, which is one of its important functions, to be a fluid membrane. And also another thing that cholesterol does, it prevents it from becoming too fluid when the temperature is too high. So we don't want the phospholipid bilayer to be either too rigid or too fluid, because both of those extremes can have a negative impact on what goes in and out of the cell. So cholesterol is really important to maintain the stability of the membrane. And moving on, we have something called a glycoprotein. A glycoprotein or a glycolipid. So glycoprotein and glycolipid are basically proteins and lipids with a carbohydrate chain attached. So the carbohydrate chain is over here. So the carbohydrate chain is found only on the exterior of the phospholipid membrane. And the reason it's on the exterior is so it can interact with other cells as a signaling molecule, and it can also be interacting with hormones and drugs. And another thing, it can act as a cell surface marker, so other cells can see the cell and identify it. So that's why it's really important that this hydrocarbon chain is on the external surface. the carbohydrate chain is on the external surface. And this is also important for sticking to cells. So in a process called cell adhesion, this carbohydrate chain can actually bind to other cells, which can be important for any transfer of substances into and out of the cell. So all of these different components are very important to forming the phospholipid bilayer, and it is really important that only safe molecules can enter the cell and prevent any molecules that may be harmful to the cell from entering the cell. So I really hope that you understood a little bit more about the phospholipid bilayer and how these different components come together to allow certain substances into and out of the cell. And yeah, thank you very much for listening. Thank you.